Here we are in Chapter 12. We're at the central nervous system. This is Part 1, and we're going to take a look at a table over here on page 429. But the first thing that I want you to know about the central nervous system is that it has parts, and the parts that we're considering here of interest is going to be specifically the brain and the spinal cord, the brain and the spinal cord. Okay, and so we start off life as a little flatworm of a microscopic nature, and we have a neural crest, which becomes a neural tube, which is filled with cerebrospinal fluid, and that's going to represent the brain and the spinal cord. There's a head and a tail end. Things begin to elaborate at a certain point. So do they tell us where we are here? This is the neural tube, and they don't really tell us time sequencing all that much here, but at any rate, we have the primary brain vesicles that develop over time, and so we have a forebrain, a midbrain, and a hindbrain. Things become a little bit more elaborate when we have actually now 5 parts of the brain. I need you to understand and name these parts: the telencephalon, diencephalon, mesencephalon, metencephalon, myel. So, in fact, if you just remember the first syllable, then you get trans, dying, and then mes, met, myel. So, telencephalon tells the story, so to speak, and diencephalon. This is the bird brain right here. Everything is reflexive and highly elaborated in terms of reflexes. Here's the midbrain, the mesencephalon, and here is the metencephalon. You could think of that as sort of the pons area, and the myelencephalon, which is the brain, the lowest part of the brain stem, the medulla. Okay, and they give us certain features right here that we can associate. This actually becomes quite almost cancerous in size. And the outer aspect here is the gray matter, where we have cell bodies of neurons, and the rest of it mostly is white matter, but we'll learn more about that soon. Okay, and then we have the flow, of course, of the canal, the fluid through the canal. So we have the lateral ventricles, the third ventricle, the cerebral aqueduct, the fourth ventricle, and then the central canal of the spinal cord. So we have these 5 parts of the brain and then the spinal cord. Okay, so I need you to have that sensibility and remember it. Now, of course, they've color-coded it. Do you see that there's some color here? Okay, so we're going to also have that same color right here. So here we are. Okay, so here we are early on in life. It says week 5. We are not yet a true fetus. We're still an embryo at week 5. So sort of on the cusp of being a fetus pretty soon. And then finally it happens for us. All right, so we're going to migrate over here to week 13. We're definitely a fetus at this point. And look at that enlargement. Holy moly. The telencephalon is becoming quite significant. And in fact, we're only going to be born with 1/3 of the structure, right? And you're going to actually produce after birth 2/3 of the cortex of your brain, as well as your children. In fact, children actually don't learn certain things early in life, so that's something that you need to sort of get in touch with and get a grip on as parents. But you can see that as things enlarge, oh my goodness, look at that. The telencephalon is really taken off there. And the gray matter is then forming into different, there's actually going to be 5 lobes per side of the brain. There's 2 hemispheres. Each hemisphere has 5 lobes, just the way you like it. So I need for you to have a sense of, this is the telencephalon, this part right here. This purple area is the diencephalon, where we have the thalamus, the hypothalamus, the pituitary. Pineal gland is in there. And we're also producing a lot of the cerebrospinal fluid in this area. that's going to be part of the system, the ventricular system. Then we have a midbrain, then we have a pons, then we have a medulla. At the level of the pons, we also have the cerebellum, okay, all forming together. So telencephalon, diencephalon, mes, met, and myel encephalon. Okay, the next thing that we take care of are the ventricles. Here's anterior posterior view of the ventricular system. We have two lateral ventricles. Do you see that? We have a third ventricle. Technically, my vice president of the medical school at San Antonio University of Texas Medical School said that the left lateral ventricle is number 1 and the right one is number 2, and this is number 3. And then we have a cerebral aqueduct. In order to get from the lateral to the third, we have to go through the foramina Monroe, which they have more recently identified as the intervertebral foramen. But I would recommend that you learn all of the nomenclature that you can, because you're going to come across people that use it. Foramina Monroe, third ventricle, cerebral aqueduct. Fourth ventricle has 4 apertures that you need to learn the names of. The foramina of Luschka are the 2 laterals, and then we're going to have a median aperture, and the things come out, and that's going to be the foramina Megendi. Megendi is the professor of Claude Bernard, who brought to us the ideas of homeostasis and the internal milieu of life in homeostasis in a particular organism. So, anyway, a very famous professor right there. Luschka was another one of those professors. Okay, so we'll come back to that from time to time, but we need to learn that system. So I'm actually going to go there. I'm going to go to the meninges first before I get to some of these other areas of the brain, okay, much as they are sweet areas of the brain. But I need you to understand meninges up front. So let's have meninges. Here they are now. Okay, so the brain is covered. You see on the outer area we have gray matter and then we have white matter inside. This is where the gray matter is, where the cell bodies are. And they're covered by a very thin layer called the pia mater, so the tender mother connective tissue layer. Okay, oops, that's up here, tender mother. And then we have the arachnoid membrane, this one right here, and it's called arachnoid because it's being held by these little denticulate ligaments. Do you see them? And they look like a spider web. And then we have a very thick layer right here. This is the Dura Mater. We have actually there's 2 layers of the Dura Mater in the brain, only one in the spinal cord. And so we have, this is referred to as the periosteal layer, and the lower one is the meningeal layer. But I would just ignore that and just say Dura Mater, tough mother. So sometimes they split apart, and we have dural sinuses. Okay, so this is an important detail that we'll come right back to. But first, we have to take a look at what are referred to as the choroid plexus right here. So this is choroid plexus. Here we go, choroid plexus. These are little capillaries, but they're not formed as you might think. They're actually formed by a combination of epithelial tissues, which are, of course, simple squamous, but most of this is actually formed by ependymal cells. So, and what they do is they transform the blood serum in certain ways and then release that transformed blood serum here into the ventricular system as cerebrospinal fluid. Okay, so the activity here, what's the source of cerebrospinal fluid? It's from the capillaries referred to as the choroid plexus. Choroid plexus. And they are present in the lateral ventricles and in the third ventricle. And then we get flow. You see it's flowing down this way through the cerebral aqueduct into the fourth ventricle, which is right in between the pons and the cerebellum. Then we have another choroid plexus. and that's going to be providing you with more cerebrospinal fluid. It's going to exit through the foramina of Meninges and Luschka, the three openings here, foramina of Luschka on either side, and the foramen of Meninges here in the center. Okay, so remember that for the rest of your life, and know that, of course, the outflow here is going into the subarachnoid space. The subarachnoid space means that your brain is actually, let me come right back here, is actually floating. You're floating inside the skull. And there's places up here at the very top in the subarachnoid space where the arachnoid capillary actually comes right straight through. The arachnoid membrane comes through, and there's going to be a return right here. This is called Arachnoid Villus. Okay, so these little finger-like projections of the arachnoid layer go in, and they release the cerebrospinal fluid to the venous blood here. This is venous blood, just the way you like it.